Hey, I'm excited to talk to you today about Curvenote and our mission towards connected publishing, where we're sharing interactive research that stays connected to the data in the notebooks, these sort of upstream early research artifacts. I'm Mike Morrison. I'm the UX lead at Curvenote, where our mission is to transform research beyond the PDF because science deserves so much better than those static PDF documents. And we really see this vision of science being open and dynamic with data and reuse built in, not bolted onto the side. The research lifecycle is messy, but for a lot of the wrong reasons. You have lab notebooks scattered across multiple different systems, different places to collaborate, and then you go to publish all your findings, and you're asked to put it all together all over again. You submit your science as this kind of glued together document, and then somebody else translates that into a typeset format. The system isn't connected at all. So our basic idea is to sort of keep that collaboration and that connectedness of those research outputs when you publish. And that's one of our visions at Curvenote, is to try to figure out how to, as best we can, connect the ideas of working and sharing so that you can go as smoothly as possible from those early insights through to the end product of publication and sharing those results. And the way we do that is through our Curvenote Reader platform. There are two ways to look at the Curvenote Reader platform. One is through the user interface, where you can upload new content directly into the system. You can see all the metadata and the policies for the article listed right here. This is kind of the back end. You can see all the files for the article. You can, of course, upload new versions, give each version a name or what changed, see a timeline of history for the article. This is kind of the content management piece, but here's the magic. You can actually submit a scientific article to multiple outlets from the same place. So we've submitted it to one place, and now we're going to add a submission to bioRxiv. The other way to look at the Curvenote platform is through these command line interfaces in action. Here are the proceedings for SciPy, the Scientific Python Conference. So here's a pull request from 2 days ago. What they've done is they're accepting all the submissions to their conference through GitHub here. And we can jump into one of the submissions and check it out. So we got some stats about the submissions, and right here is really cool. We have these checks where when you submit to the conference, we can check for things like maybe you didn't include a DOI with one of your references and we flagged that. Or maybe you did include an ORCID ID, so you get a green check. Now, here's the magic part. Authors can actually preview the typeset draft right after submission. This kind of flips the submission process upside down, where you start with this pretty typeset draft instead of ending with that. And then you can just improve on it. So in this case, the author submitted a Jupyter notebook, and we're rendering it here with all the code snippets, these big, beautifully sized figures. We've got the equations with the code snippet that created them. So it's the Jupyter notebook rendered directly in Curvenote. You can also, if you want a PDF, you can still get one. We just generate one immediately. And it looks pretty, but still a PDF. So these are some of the systems that Curvenote sees that enable us to get a little bit past the sort of reading and access era where you just read this static document to experience science, and towards this new utility and reuse era where you have all the data and the code and the methods bundled together much more usefully. So you can kind of stand on the shoulders of giants faster. You can reuse the code more easily. You can get to the data more easily, and it's all there for you in a much more interactive way than it's been before. We're making every single aspect of a scientific study easier to reuse. You can reuse the data more easily, the code behind the figures. You can sort of fork them and make your own version of the figure or apply it to your own data. You've got your methods much more easily. The idea is to help people extend previous studies faster, so you can kind of stand on the shoulders of giants more easily. Whereas a lot of times with a PDF, you're kind of like, the American expression is squeezing blood from a stone. You're just trying to get this like static text to do something for you. It's a lot harder to work with that static text and tiny little figures in a PDF. And when you have this interactive publication where each piece of the scientific study is easier to reuse and more flexible, it can actually communicate a lot better too. I'm going to show you some of those possibilities. How we can improve the experience of learning science when we connect that upstream data and move into more interactive ways of publishing content. Maybe that's interactive charts. In this case, you're scrolling through earthquake data over time. Maybe zoom in on the time period that you're studying. You could click a button and spin up computational resources. In this case, it's a Jupyter server behind the scenes. You're showing all of that being fully reproducible directly in line, so that you go from talking about a model to something that's fully reproducible. Or here's an example of a rat running around a maze with an implant in its head, reading its brain to try to figure out actually where it thinks it is. And there's many hours of time series behind this. You can flip through these events and actually see and interrogate these results. We can also take pictures that used to have to be static, Like here is a 1.5 terabyte image of a zebrafish. It's a microscope image, so it's huge. And typically what you do in traditional scientific publishing is you'd kind of like screenshot it and then include a little zoom box like that. And that's the best you could get because you're limited to these static images. In this case, our partner, Elemental Microscopy, came to us with this problem. How do we actually put these huge terabyte-sized data inside of our articles so that readers of our journal can actually browse and explore the zebrafish image or whatever? So we're working with some standards in this space, OME-ZAR files specifically, to be able to zoom in directly into that image inside of the article, inside the reading experience, in line. You're not just screenshotting this, you're exploring the cellular structure of a zebrafish right inside the article. So we can demonstrate and integrate all of this content together. The SciPy Proceedings is another place where we can go directly in and start to show visualization content in different ways. And so in this case, this article was about a unique visualization technique for transparency. And we can show that directly in the browser. We can actually show the research in action. In this case, dive into that content and experience it. And so these are sort of a smattering of what we see as connected publishing. Connecting directly into the content, being able to connect the data, the code, the environment, the software, and pass that all the way through into the ways you share the scientific work. And that unlocks some new possibilities. But you've probably seen demos like this before. Maybe not this cool, but you've seen demos of interactive computational articles before. The big question we often get is, will it scale? Like, I'll start getting excited when you can scale this past one journal or past one article, because that's when things actually change. Well, it scales. So the second piece I'm really excited to announce today is a new partnership with openRxiv. This was announced just a few days ago on June 1st, so I'm going to show you a sneak peek of what's coming. Curvenote partnered with openRxiv through openRxiv Labs to launch a new reading experience across all of bioRxive's content, all of Medic's content. We've converted over 500,000 preprints to this upgraded, modernized reading experience. That's over 34 terabytes of scientific studies that we've now reprocessed into a new reading experience, and that's now available at reader.openarchivelabs.org. So I'm going to show you a little bit of a preview of that. Behind the scenes, it looks like this. So this is what bioRxiv sees. We have our scientific content management system that's running and hosting all of bioRxive's preprints. So you can search them, filter, see the history of them, jump into the details. This is a project called Spyglass, a preprint of one of the authors that we've also worked with, showing all their past publications. And then we can jump into the front-end experience. So we start with a basic list view. You can see this if you go to reader.openarchivelabs.org. You can filter and search by different time periods as a start, or jump into different subject areas. Now let's look at the reading experience. You get this beautiful reading experience that we paid a lot of attention to in terms of the design aspects. You can hover over different abbreviations and see the definition expanded, which is a really, really underrated feature for saving you from scrolling up and down to find where a term was defined. And again, that's across all preprints. You can switch the mode to dark mode. You can hover over references and it'll pull in the abstract for that reference. You can get a quick overview of a study that's being referred to without losing your place. You have really generously sized figures. You have figure references. So this is showing figure 2 right next to figure 2, but imagine, a reference to a figure if it's 5 pages away, you can kind of hover it and see the figure in the box. You can also lightbox like this. You can zoom into figures. So this has been a massive effort that the Curvenote team has been working on with openRxiv. It was a full visual redesign that uses the Curvenote Reader to reprocess and upgrade JATS content to enable new experiences. It includes metadata improvements like citations, abbreviations, figure cross-references, and it opens new possibilities for computational content. And again, it's officially launching June 8th, which by the time you see this video should be already passed. And let us know what you think so far. Again, it's reader.openarchivelabs.org. Now I'm going to show you a few more of those user interface improvements that I, as UX lead at Curvenote, am super proud of. Again, you can hover over references to figures and see the figure, which saves you from scrolling. And this part I'm really excited about. You see how it's about to hover over this reference to supplementary figures? When we hover it, we display the supplementary figure. So a lot of times scientists will put really incredible scientific images in their supplement section because the journals don't really restrict format in supplements like they do in the main article body. So we find these gorgeous microscopy videos and things like that just sitting in supplements. Now, when you reference a supplementary figure in the article, we go dig it out of the supplements and show it in the pop-up. So we're starting to unearth all of that buried scientific treasure in the supplements in these articles. And again, this is across 500,000 preprints right now. So there's really no telling what kind of new things we might stumble across now. We're also working with protein databases, so we can try things like maybe you hover a protein name and we show an explanation of the protein right in the text. All of these things that a lot of scientists in the same field have to deal with regularly, we can start standardizing so that people don't have to redo the same work as often. We're also working to interpret tables. A lot of tables in bioRxiv preprints are published as like images or pictures of a table, which is kind of hard to search and browse. We're working on interpreting those and rendering them as actual tables. This is all really just the beginning. And one of the best parts about this is that all of these amazing features are made possible by structured information behind the scenes. You can go to any Curvenote Reader article and add .json to the end of the URL, and you can see the full JSON structure of the article and access any single piece of it programmatically. What this means is that these Curvenote Reader articles are easier and more fun to read for us humans, and they're easier to read for robots. They're more machine readable. Some of you are wondering, well, maybe I'll get all my science through AI chat in the future and we won't even need articles. What this allows for is for the AI chat to start pulling in pieces of scientific articles verbatim, not overinterpreting them, into the AI chat stream. This kind of machine readability allows for that. And this machine readability of scientific articles is moving towards something called the openExchange architecture. It's a new standard for modular and composable scientific content. And there was a talk last week at Force 11 by Tracy Teal of openRxiv and Nakomi Bentley of Stencilla about this openExchange architecture. So expect to hear more about this in the future. We're really happy at Curvenote to be able to do our part to make science more modular and expose different pieces of the scientific study in these different ways. And we're especially excited to scale all of this with our partners using these open science standards. So we can connect research from its origins, where it starts in the lab being collaborated on, to where it's shared with everybody else so they can build on it further. Thanks for watching. Again, I'm Mike Morrison. I'm UX lead at Curvenote. And if you have any feedback about the Curvenote Reader, just please email me. I'm ux@curvenote.com. And here's that URL again so you can try it for yourself.